Okay, so good morning, everyone. So first of all, I want to thank the organizers for inviting me today. I'm really happy to be here and grateful. So as Rafael said, our team is part of the Laboratory of Computational and Quantitative Biology. Here, is it better? Yes, thank you. Here, so that's good. Okay. So our team is part of the Laboratory of Computational and Quantitative Biology, which is part of the Institut Biologie Paris-Seine, Sorbonne Université, and we're located on the Jussieu campus in Paris. And so our research activities. So research activities revolve around the sequence, structure, function relationship of biomolecules, and in particular proteins. So we've been developing predictive, scalable, and interpretable algorithms and computational methods for deciphering the complexities underlying this relationship. So in particular, we've been addressing the multiplicity of protein partners in the cell, the multiplicity of protein functional states, and the multiplicity of proteoforms, protein isoforms that are generated by alternative splicing. So today, I will tell you two stories. The first one is about assessing the functional impact of mutations on protein function. And the second one is about protein dynamics and motions. And in both cases, I will show how and to what extent protein language models can help us for these tasks. Okay, so let's say we have a protein that contains a certain number of amino acids, say L. So each of these amino acids can be substituted by 19 other possible amino acids. So what we would like to do is to be able to estimate the effect of all of these possible mutations, so that's 19 by L, on the functioning of the protein. So you can provide an answer to that questions with experiments through deep mutational scans. So you have here a sketch of the experiment. So you start from a library containing the wild type and mutants you're interested in. And then they go through a selection process that corresponds to the phenotype that you want to measure. And that can be organismal fitness, that can be binding to a partner, or that can be folding stability. And then you count the proportion of the different mutants that you have after the selection. So these experiments have been conducted on a couple of hundred proteins, and these experimental data are available or compiled and curated in the protein JOBIM benchmark, which is developed by David Mark's lab. So currently, the benchmark contains over 2.5 million variants from different taxa, human, bacteria, viruses. So that's a lot, but at the same time, we're very far from being able to experimentally assess entire proteins, screen entire proteins. So this has stimulated the development of computational methods to do this task. In the protein JOBIM benchmark, you also have a leaderboard where you can see different methods, their performance, and they are basically evaluated all in the same way. So in recent years, we've seen a rising interest in computational methods that are weakly or unsupervised. And why is that? Because these experimental data contain some noise and some biases, and they're quite sparse. So you have a list of methods here that I put some historical method and some more recent method. It's not an exhaustive list. So what these all of these methods do is that they exploit protein sequence information across different species, so across evolution. Some of them also use population data. So they look at the polymorphism within human populations, for instance. So these are the methods that I highlighted in blue. So you may think that accounting for this polymorphism can be beneficial if you want to predict, for instance, the pathogenicity of mutations. But in fact, there is a recent study by Joe Marsh lab showing that the predictors that do not use this population data perform more or less the same as those that use them. And that when you use population data, you have strong biases towards specific populations. So for instance, European versus African. And we also have some methods that are a bit more sophisticated and integrate structural information. So for instance, alpha-missense that was developed by DeepMind uses population data, sequence data across species, and also supervision on 3D structures. So in this landscape, some years ago, we developed a method which takes as inputs a multiple sequence alignment where you have the query sequence, the query protein at the top, and it produces a mutational landscape. So that's exactly what I was talking about at the beginning. So we have all the possible mutations, and basically we have a numerical value saying if each mutation is impactful or neutral. So JEM is not a machine learning based method. It has an explicit model of the evolutionary relationship between protein sequences, and it has very few biologically meaningful parameters. So I'm not going to describe in detail the method, but just the two main ingredients. The first one is a measure of conservation that accounts for the full context. So I illustrate this with this tree here. So you have a bunch of sequences, and now we're going to compare the position on the left, position 3, and position 13 on the right. So if you look only at the frequencies of occurrence of acids at this position, you will say that position 13 is more conserved than position 3. Why? Because you have more Ks basically here than you have Fs here. But now if you account for the topology of the phylogenetic tree that relate these sequences, you can see that for F, you can go further up the tree than the amino acids that you have at position 13, okay? So basically you have a bigger subtree that conserves F. So this is what we do. We account for this topology. And so we're going to say that position 3 is more conserved. And because the tree is built based on global similarities between sequences, we have an global sequence context when we measure the conservation of each position. And the second ingredient is a notion of evolutionary distance. So what we do if we're interested in a mutation from A to G, we're going to look at natural sequences that have the mutation. And we look at the closest sequence that has this mutation. So we compute this evolutionary distance, which represents the minimum amount of changes that you need to accommodate the mutation. And so intuitively, the more conserved the position, the more sensitive it will be to mutations. And the bigger the amount of change you need to do for a specific substitution, and the more impactful the substitution. Okay, so GEM is one of the best predictors according to the protein GEM benchmark. It has been used in different studies. So I show here some works by Preston Lindorf Larson lab. So here what they did is they looked at they related the scores from GEM, which are purely based on evolution and sequences, with predictions for folding stability changes. And so by comparing those, you can provide mechanistic insight into the effect of mutations. So with GEM, you will have a bunch of mutations that are predicted as deleterious or impactful. And some of them is because they really disturb the folding of the protein, so the stability. But others is because they will impact the function in some other way. So you can kind of disentangle these effects. This has also been done in a recent study where they set to massively measure these folding stability changes. And we've been also using it recently in a work where we developed a geometric deep learning method for predicting the impact of mutations on binding affinity. Okay, so as I said at the beginning, GEM is, I forgot to mention, but you can use GEM. It's available as a standalone package, web server. I encourage you to use it. So it takes as input multiple sequence alignments. And so initially, this was kind of a bottleneck for the computational efficiency because it could take, so we were using Jacmer profile HMM method, and it could take more than 1 hour for a protein. So when AlphaFold came out, Sergey Ovchinnikov and Martin Steigenegger lab developed ColabFold to make it accessible. And so they devised a strategy for generating the multiple sequence alignment that is very cheap, very fast, and that aims at maximizing diversity while minimizing the number of sequences. So it's based on the MM6-2 algorithm. And we realized that this strategy is also beneficial for the prediction of the effect of mutation. So we kind of systematically assessed several protocols for generating multiple sequence alignments. So here I show two comparisons. We have colorful, and then we have JACMR, which is profile HMM based method. So basically it's more sensitive, the profile HMM. So you will get more sequences of the order of 500,000, where you have 20,000 with colorful. In terms of performance, you roughly obtain the same thing. So how do we measure performance? It's the Spearman rank correlation coefficient between the metrics that we predict and the metrics that is the experimental metrics. So we're looking at how well we rank the mutations. So you can see here that the performance is roughly similar, except for these two outliers. What's happening here is that we have viral proteins. So for these viral families, differences are very close to each other. The variability of the input MSA is very low. And basically, colorful reduces this redundancy. So in the end, you have very few sequences. But this is very So this was an important finding for us because it means that now we can have these predictions at a much cheaper cost. And so we can run predictions for entire proteomes within a couple of days. So we did this for the human proteome, and this is available to the community. We did this also for the fly proteome. So I will not describe these results, but Marina is currently looking at the genotype-phenotype relationship in fly and relate the force to organismal fitness. So I encourage you to go check her poster, number 128. Okay. That we can generate in a reasonable amount of time, and they are fully interpretable. So we know the algorithm that generates sense they are homogeneous. And they are good approximations of the experimental measurements. So we wondered whether we could use them as surrogates for experiment. Capacity transformers that are trained on huge row sequences to predict, to reconstruct masked amino acids. So just like natural language models, reconstruct words in sentences. And so while they do this, they learn a representation sequence vector for each amino acid, about 1,000 numbers. And these representations, which we lumps, contain some information. So basically, if you project them back into a low-dimensional space, you can see that you can segregate proteins by function, by their evolutionary relationship, by their physical chemical properties. So these protein language models are expressive enough to be applicable to a broad range of protein-related structural information, functional annotation. So I guess it's really interesting to look into this. So we trained the model on 5,000 human proteins, and then we assessed it on the protein GYM benchmark. So here I show the results for, we have humans, we have other eukaryotes, prokaryotes, and we compare to these 4 methods here. So basically we have the teacher GEM, we have the baseline, it's just the protein language, 0 shots. And then we took the top ranked predictor at the time in protein GYM, and the best method that is purely based on protein language model, VESPA. Okay, so what you can see here is that VESPA is performing well. Basically, it matches state-of-the-art prediction, so it's roughly similar to the teacher GEM. And then you have a significant increase over these 3 bits, and that's on 161 proteins. So we can also look at the different phenotypes that are measured. So we have a variety of phenotypes. What's kind of interesting here is that you see that the methods all kind of perform bad on the binding phenotype. So there is clearly here a margin for improvement. And more or less we have same picture. And then what's happening if we look at viral proteins? So I told you the viral proteins, the language models, all of the language models do not represent them well. So in fact, we see that here we have indeed a decrease in performance compared to GEM and Transcept-Eve. But if you compare with the ESM2, vanilla ESM2 zero-shot prediction, we improve a lot on this. So that means that the supervision on GEM is able to partially compensate for the poor quality of these viral protein embeds. So now we can look at a bit more methods. So again, it's viral proteins, and I'm showing here other version of ESM, and also a recent protein language model, SAPROT, that uses structural information. And you can see that basically all of them perform that. Performance is much lower than the other method, and in particular than VESPA. So we looked a bit at the predictions in details, and we have some interesting cases like this one. So this is the yeast ubiquitin, and this prediction kind of challenges the common view that strong evolutionary pressure will imply high mutational sensitivity. So basically it has a lot of positions that are not sensitive, that are very tolerant to mutations when you look at the experimental measurements. And so this is a case where the predictions from VESPA agree with the experiments better than GEM, like substantially better. So I kind of highlight here where there is disagreement between the methods. So in particular, you see you have these glycines at the end that are very red, so meaning that they are intolerant to mutation, they're sensitive to mutation, and they're predicted as sensitive to mutations by VESPA, while GEM predicts them as tolerant. So we kind of misses here the points. And this is the opposite for this glycine 35, where you see it's neutral here, and here it's predicted as highly sensitive. So it's just one example. It's kind of difficult to assess this quantitatively, but the intuition is that we have some kind of regularization of GEM scores by VESPA. So another thing that we looked at is how many sequences we need for training and what changes when we change the training sets. So you have here different experiments where we train on a few thousand proteins from just one organism. So we have human, we have fly, we have E. coli, and then we have viruses, which are many viruses. So what you can see is that basically the prediction performance are pretty stable across this different training regime. So our baseline is, our default is just train on human, and you can see that we can then, so here you have the different viruses that correspond to the different taxa. So this is prokaryote, eukaryote, human, and virus. So you can see that we can generalize from human to prokaryotes to other eukaryotes. We can also generalize from fly to the other taxa. We can generalize from E. coli to other taxa. Basically, the performance is always the same. We have a slight degradation of performance when we train on viral proteins, but it's not that much. So it means that the mapping that we learn just by training on viral sequences is still valid for the other taxa. It's still transferable. But then we struggle for achieving good performance on viruses at the test time, at inference time. So whatever here the training set that we use, even if we train on viruses, we don't really improve over the other training regime. And another thing that we see is that we don't need more than a couple thousand sequences. Basically, if we take all the union of all these training sets, we don't have improvement in performance. One advantage of the method is that it's super fast. So it's orders of magnitude faster than GEM and Vespa, which are themselves orders of magnitude faster than the other predictors. So you can basically predict all the mutational landscape of the human proteome in 30 minutes on a laptop. And then so in the future, we can think of, if the protein language model representation improve, in particular for viruses, we can just apply this scheme to other embeddings. It's quite straightforward. Okay. And so this was a work done by Sabine Marquet and Julie Schlenstock, who were part of the Burkett Ross lab, with whom we collaborated on this, and was one of the first academic lab to develop language models. So lesson learned and challenges. GEM evolutionary scores can effectively serve as surrogates for experimental measurements. We can map directly these PLM embeddings to the mutational outcome. So this is different from what was done before, because here we do not compute these log-odd probability ratios. So it's just a mapping function between the embeddings and the output. And so we just need a couple thousand proteins to have high quality prediction. And then we have this kind of counterbalancing effect for the poor quality of the embedding and the regularization of GEM by Vespa. So it kind of takes the best of both worlds. Also, although the predictions are not really impressive. Okay, so for the second part of the talk, I'm gonna move to protein motions. So why are protein motions important? Because they determine the way protein interacts with their environment and with their partners, and so they determine the way protein function. So here you have conformational transitions, functional transitions that were determined by a combination of experimental and computational techniques. So the first question we asked ourselves here is, how can we describe and visualize this conformational variability that we have in protein structures? And for this, we developed DANCE, which is an automated pipeline that starts from takes as input a set of 3D structures. So these can be experimental structures or models, for instance, computed by AlphaFold. So you have 3D coordinates for atoms, and you also have the amino acid sequence. And so what DANCE does is that it clusters the protein chains in the input structures based on their sequence similarity. And then it aligns them within each cluster, again, based on sequence similarities. So this can be basically adapted or bypassed to use structural similarity, but by default sequence. And then it will superimpose the conformations that we have in these clusters to generate these conformational collections. Okay, so that's what you see here. So we applied it on the whole protein data bank. That's over 700,000 protein chains. It took about 2 hours, 16 CPUs. And what you can do is you can play with the level of sequence similarity that you use here to generate your clusters, to segregate your sequences. So we looked at how the conformational collections evolve when you change the threshold. So basically here you have the gain of conformation when you go from the most stringent setup, 80% sequence identity, 80% coverage, to the most relaxed one, 30% sequence identity, 50% coverage. And basically we see the biases that we have in the protein data bank. So we have a few protein families, for instance, kinases, proteases that are overrepresented. So basically when you relax the threshold, you get all this remote homologs into one conformational collection. So you have a huge expansion. And for most of the collections, you only have a gain of a few conformations. Okay, so what do we do with these collections now? So here you have the example of adenylate kinase. So on the left, you have the 42 conformations we have in the collection. It's a stringent setup, so 80% sequence identity and coverage. So we take the 3D coordinates, we compute the covariance matrix, and then we decompose this covariance matrix, and we get principal components, which are the eigenvectors of the covariance matrix. And so I illustrate this first principal component here. So basically, it defines a set of 3D displacement vectors for each atom in the protein. So here we're working only with C alpha atom. So you see these yellow arrows that correspond to the first component. And then you have the eigenvalues that tell you how much of this, the total variance is explained by each component. So for instance, in that case, we have just the first principal component that explains 99% of the total variance. It means that the variability that we observe here is explained by this principal component. And so we interpret these components in terms of linear motions. So what you see on the right is basically deformation of the protein along this component, and it corresponds, you can see it corresponds to the functional transition of the protein. So with this, we can now estimate the motion complexity that we have in our collections. So we look at how many linear motions we need to explain most of the variance. And okay, we set the threshold, most of the variance is okay. So we do this across the different sequence similarity thresholds. This is what you have here. So different versions of the collections, they have different size, they have closed homolog, remote homolog. And you can see that basically this picture is very stable across these different levels. So what we see here is that for half of the collections, we need only one or two principal components to explain most of the variability that we have. And for most of the collections, and there is no automated policy that we can apply to set up these parameters. So it limits the practical usage. And what we observed is that the reconstruction accuracy really depends on the distance of these test points to the convex hull of the training set. So basically, if we are in the vicinity for angstrom, we get accurate reconstructions. And then if we go further away, then the reconstructions are much worse. So there is generalization capability, but really in the vicinity of the training set. So now we may wonder, beyond the accuracy, the RMSD, do the conformation look good? Do they look like proteins? So for this, we looked at the proportion of residues that we have in the most allowed regions of the Ramachandran plots. So that's an indicator of the quality of the structures. And here you have the different flavors of KPCA. You also have UMAP PCA, and the ground truth is in yellow. So what you can see is that for some of these proteins, you have basically the quality of the reconstruction is more or less the same as the quality of the ground truth structures. What you can also see is that UMAP performs very bad. So I didn't show this before, but also for the RMSD reconstruction accuracy, we get very, very wrong results. Very high errors for UMAP. And some kind of interesting observation here. So we have in this collection for RAS a experimental structure that is of poor quality. Okay, so you can see here the secondary structure are not well defined. We have only 64% of the residues that are in allowed regions. And basically, when we reconstruct the structure, we get a much better quality, over 90%. So what's happening here is that we have a kind of denoising effect of the PCA. Thanks to the conformations, the other conformations that are in the collection and that have high quality. Okay, so yeah, just a transition. So what I showed here is predicting unseen conformations from known conformations, okay? So we start from 3D structure information and we predict new conformations. But then we have this description of motions in terms of principal components. So there's a continuous representation of motions. So what we recently tried is to start from sequences, sequence information only, and to predict the motions, not conformation, not sampling conformations. So this is the framework that you see here, kind of looks like VSPG. So now we call it CMON. And it's a bit different. So we start again from the structure. We compute structure, not sequence. We compute an embedding with a pre-trained language model. And then we are going to apply what CMON does basically is that it applies 1D convolutions to the embeddings. So we have just 2 hidden layers with a kernel size of 15 and 31. So that's rather small. You have just a local neighborhood of each residue in the sequence, along the sequence. And then CMON predicts an arbitrary number of motions. So what are these linear motions? They're exactly the same object as the principal components. Okay, so they define a set of 3D displacement vector for each atom. And this is what I illustrate here. But again, we only predict the arrows. We don't have the knowledge of the structure. The structure is not here. And so during training, what we're going to do is that we're going to regress these motions on the principal components that dense extracted from the experimental structures from the experimental collections, okay? So what I also show here is 2 predictions just to give you a sense of what's kind of acceptable and what's not. So we have this normalized sum of square error that we use to assess the quality of our predictions. And basically, when we inspected the prediction, we could set a threshold of 0.6. So roughly, if you are below 0.6, the predictions are acceptable. So you can see here, it more or less matches the yellow one, which is the ground truth. And then the red one, which is much higher, is completely off. It's not completely off, but it's much worse. Okay, so here we used 2 pre-trained language models, ESM2, 3 billion parameters, just like before for VSPG, and also PROST-T5. So PROST-T5 is a bilingual model that was trained to translate amino acid sequences into sequences of 3DI tokens. So 3DI tokens are just a structural alphabet that is used by the structural alignment method FOLSEQ. So basically, you train the model to translate sequences into one-dimensional representation of structures. Okay, so you train it on the AlphaFold models. So it has some structural information. So we train CMON on 5,000 to 15,000 information collections from the PDB. And this is basically the kind of summary of the results. So what you see here is the a number of proteins in the test set. So we have about 1,000 proteins in the test set for which we predict at least 1 motion. So I should say that we predict 3 motions and we regress over 3 ground truth motions. And here we look for how many proteins for which we have at least 1 prediction that is better, that has a smaller error than what you see in X here, okay? And then, so I have the comparison between these 2 embeddings. So ESM2 is in blue, and then PROST-T5 is in orange. So what we can see is that we have significant, substantial improvement when we use embeddings that are structurally aware. And we also compare the methods to the normal mode analysis. So here it's not based on machine learning, it's physics-based. It takes as input a 3D structure. It builds an elastic network from this 3D structure, and then it decomposes the Hessian of the potential of this elastic network. So what we get, these normal modes are basically the motions that are energetically accessible. So it's purely structure-based. So you have a Cohen continuum, we have ESM2 purely sequenced, then we have some PLM that is structure-aware, and then we have the normal mode analysis, which is really structure-based. Okay, and so you can see that we predict better than the normal mode analysis. It's a bit subtle here, but then for cross T5, it's significant. And then we have two curves because we did some data augmentation with two details, but I can develop after. Okay, so if we look at the individual predictions, that's what we have here. So you have the loss, the errors for Simon ESM2, Simon cross T5, and the normal mode analysis is the color. So you see that basically the three methods kind of agree in the sense that the best predictions that we have an overlap of best predictions for the three methods that's significant. And they also agree on what they miss. So we have here some kind of doubts also on the ground truth. So in our collections, we have sometimes insertions of segments that provoke some motion, which is not really biologically significant. We have some extremities that move. So there is also some question on a number of collections about the biological significance of the motion. Okay, so for illustration, we can pick up one guy here. This is the Legionella effector MAVE-C. So you can see here the arrows for the ground truth components, and then you have the predictions. So in that case, you really see that the error is much lower for PROS T5 than ESM2. This is visible here. These orange arrows match the yellow one, while the blue one are much kind of off. And also the normal mode analysis completely misses the motion. Okay, so in that case, the this test protein has no sequence similarity to the training set, and it has a weak structural similarity. So you can see this is shown here. The color corresponds to sequence identity to the training set, and in the x-axis you have the TM score, which reflect the structural similarity. So the TM score is below 0.6. So basically, it has some structural similarity, but it's not exactly the same fold in the training set. So it means that we have some generalization capability to proteins that are kind of far from the training set. So if you look at the results here and the points, you can see that there is a trend. So obviously the best prediction we get here, they have high sequence identity, they have also high TM scores. But then we still have low errors for proteins that have no sequence similarity and low structural similarity. So you have here the Pearson correlation, basically. So that's kind of okay. In terms of time. Okay. Okay, so if we look now a bit in details at the comparison between the normal mode analysis and the predictions that we get from Simon. So if you take the top 100 proteins best predicted by Simon, about a third have motions that are completely missed by the normal mode analysis. So that's kind of interesting because it means that we have motions here that you cannot infer from the 3D structure. They do not seem to be geometrically inferable. So here are some examples of this. So you can see these are two bacterial toxins. So on top you have this motion of, and all of these are functional motions, okay? So this I verified in the literature. So you have this loop motion here that is well captured by Simon, but then the normal mode analysis is completely off in terms of what region moves. And so in that case here, you have a huge conformational change. So there is this whole Sion part that goes like this, and this is for like fusion process with and forming aptamers that are going to be aptamers for the membrane. And so again, we have a good prediction with Simon, and this is in blue. So this is ESM based. There is no structural information in the embeddings here. Well, for the NMA, it's not so bad, but it has this kind of breathing motion. So here we see also the limitation of normal mode analysis, which is expected. So basically it's in elastic network models. So if you have a motion that involves a large rearrangement of secondary structure, you will not be able to capture this. So it's kind of interesting to see that maybe we could combine this method because there is complementarity in the predictions. So again, no sequence similarities to the training set, but there are some structural similarities. So we have likely transfer here of other proteins that have the same shape and have the same function space, which is also interesting to notice. Okay, so to wrap up this part, what I showed is going from conformational collections to motions by extracting the principal components, and these conformations lie on the low-dimensional manifold. On the predictive part, so we did two things. We started from some subset of conformations and predicted other conformations by going through this low-dimensional representation. And starting from sequences to go directly to motions with protein language models. And we have, so there's limitations in generalization and complementary. Okay, so just to finish, I want to advertise a bit. So I already advertised Marine's poster. So again, if you want to check it out. We have also two posters from Louis and Julien on the part I did not talk at all about, our work on alternative splicing. So if you're interested, go find them. So this is more like deep learning and evolution. I'm also advertising for Arno Leerman, who is joined us as a postdoc in the team and is going to present his work during his PhD about segmentation and differential analysis. We have a mini symposium on Thursday, but I guess you need to be registered probably, but still on advertising. So it's going to be about machine learning and statistics and genomics and metagenomics. And so I'd like to thank the people who did the work, Julius, Céline, Birkat, Marina, Alessandra. Also, we collaborated when we developed GEM. So this is the mutation part. Valentin Lombard is a PhD student in the team, co-supervised with Sergei Gridinin on the protein motion parts and some other members of the team and collaborators. And I want to thank you for your attention.